way. And today I'm going to start by telling you, if I can get my slides to advance, what I think are some of the key challenges in human genomics research. So the first is that we need to do a much better job at characterizing both genomic and phenotypic variation in ethnically diverse populations from across the globe. We need to understand the evolutionary processes that generate and maintain that variation, and to better understand how gene-protein, and gene-environment interactions contribute both to normal variation and disease risk. As you know, as you mentioned, the focus of my research is on Africa. And I've been focused on Africa really since the beginning of my career. When I was at Yale University, it was when I first started, this was way back when, and we were looking at variation in the nuclear genome, and notice how much variation there was between the different African populations compared to non-African populations. And that got me really interested. And then had come to, for a short visit to WITS, working with Prof. Trevor Jenkins, I hope I'm getting that right. And that again just got me even more interested. And so it's been a passion ever since. And one of the reasons is that I actually have a background in anthropology and human genetics. And these red dots are showing the locations of fossils of anatomically modern humans, the oldest of which is dated to 300,000 years ago. So we know that modern humans arose in Africa. And I was always really interested in learning more about African population history and human evolutionary origins in Africa. And then, as you know, somewhere, let's say, between 50 to 80,000 years ago, relatively small numbers of people migrated out of Africa, giving rise to populations across the globe. And after they left Africa, they were admixed to some small extent with archaic populations like Neanderthals and Denisovans, such that now somewhere between, say, about 2% to around 6% of the genomes of people from outside of Africa are derived from these archaic populations. Now, it's very likely that this was happening in Africa as well. There's no reason to think that it was any different. There may have been a lot of population substructure and some populations that had very diverged lineages, and there could have been small amounts of admixture. It's just that it's much more challenging to distinguish because we don't have ancient reference genomes in Africa because the DNA doesn't preserve as well. But I think it's just a matter of time before we will. The other thing that motivates my research is trying to understand risk factors for health disparities. So for example, in the United States, people of African ancestry have higher prevalence of hypertension. People of Native American ancestry and African ancestry have relatively higher levels of type 2 diabetes. There are, without doubt, major social, demographic, economic factors that are influencing these health disparities, and including things like access to healthcare and, frankly, structural racism that exists in the US. And that may, in fact, that probably is the driving force behind these differences, but it's thought that there may also be genetic risk factors as well. And the biggest challenge is, how do we distinguish the genetic from the environmental factors that are influencing disease risk? So in this perspective we wrote a few years ago, we were looking at the GWAS, the Genome-wide Association Study Catalog. And as of that time, roughly 80% of the individuals being used in GWAS were of European ancestry, about 10% of Asian ancestry, 2% African ancestry, 1% Hispanic ancestry, and less than 1% everybody else. And if we look at other commonly used databases like Nomad or GTEx, again, you see this major European bias. That's really a problem because it's going to impede our ability to fully understand the genetic risk factors of human disease, and it could actually exacerbate health inequalities. Our ability to translate genetic research into clinical practice and public health policy could be incomplete, or even worse, it could be mistaken. It could result in inaccurate assessment of pathogenic variants in clinical genomic studies. And one of the reasons for that is that one of the criteria for determining if a variant of unknown significance is pathogenic is how common it is, with the idea being if it's very common, it's less likely to be pathogenic. Well, maybe it's not common in European or Asian populations, but it's common in African populations. So then you might actually change your assessment of that impact of that variant. And then lastly, attempts to use estimates of genetic risk from European-based studies in non-Europeans may result in inaccurate assessment of risk and lack of appropriate interventions. I think many of you have probably heard of polygenic risk scores, but just to remind you that a polygenic risk score is calculated from the weighted sum of the effect sizes of the genetic variants used in a genome-wide association study. So for example, if we're looking at, say, risk for heart disease, people who have a lot of the alleles or the variants that are associated with high risk for heart disease, if you add up all the different variants, they're going to be at the high end of the tail of the distribution. They're going to be at much higher risk for heart disease. People are using this right now in the clinic. They're using it to predict who's at risk, maybe even to do preventative measures or to determine therapeutics and things like that. But in this nice study that Alicia Martin and colleagues had published a few years ago, they showed that when you calculate a polygenic risk score in Europeans, it does really well at predicting the phenotype in Europeans, but it doesn't do so well when you're looking at other ethnic groups, and it does particularly poorly in people of African ancestry. And there are likely to be a number of reasons for that. But one of which is the fact that, as you know, when we do a genome-wide association study, rarely do we actually capture the causal variant, or we don't know what the causal variant is. So you're identifying a variant that is in linkage disequilibrium, or it's associated with the causal variant. Now, because of that demographic history I showed you, with a small set of people leaving Africa and going outside of Africa, when that happened, you see more extensive linkage disequilibrium outside of Africa. And you see more variation within Africa and actually between African populations. So what we call a tag SNP, a variant tagging a causal, the SNP carrying tagging a causal variant may differ in different populations. So it may be exactly the same causal variant, but you just need to find better tag SNPs. And there are some approaches for doing that. But the other possibility is that there could actually be different causal variants in the different populations, possibly due to their demographic and evolutionary history and adaptation to different environments. And if that's the case, we're not going to find them unless we look in ethnically diverse populations. So I talked about these first two. And the third is that there could be different effect sizes in different populations due to differences in allele frequency, gene by gene, and really importantly, gene by environment effects. So there's a lot that we need to do in terms of better understanding how to calculate these risk scores and to transfer them across ethnic groups or different ancestries. Okay, I think everybody here probably is familiar with this, but just to remind you that the languages spoken in Africa, there are over 2,000, have been classified into 4 major language families. So people who speak Afroasiatic are mainly in the north of Africa and the eastern part of Africa. People who speak Nilo-Saharan languages, shown in red, are typically in central and eastern Africa. People who speak Niger Kordofani, now most people say Niger Congo, are shown in the sort of yellow-orange color, and they originated in Western Africa. The largest subfamily are the Bantu languages, which are thought to have originated around the border of Cameroon and Nigeria. And people, the Niger Congo speaking people had very early on developed iron tool technology. and what's called slash and burn agriculture. So they were really effective at going into the tropical forest, cutting down the woods, growing crops, and being able to sustain large population sizes. And so in the past, say, 4,000 years, there was a massive amount of migration from that region to the east and to the south of Africa, and then to the west and to the south of Africa, where they then interbred with some of the local population. So it's really shaped the, I would say, genomic landscape in Africa. And then the last language family shown in green is Khoisan. And that includes some of the San populations in southern Africa who speak with clicks, and until recently they practice a traditional hunting and gathering lifestyle. And then in Tanzania, there are two groups called the Hadza and the Sandawe, who also speak with clicks, although it's been really contentious. Some people say that their languages are so different, they really shouldn't even be in the same language family. And the Hadza continue to practice a traditional hunting and gathering lifestyle. The Sandawe have admixed with some of the neighboring groups and have adopted different subsistence patterns. So this work wouldn't be possible without these different partners that we've worked with in Africa, some of whom are shown here. I'm sure some of these people are familiar to you, but this really is a partnership, and a lot of, as I'll tell you in a moment, a lot of what we do is also training, and we want this to be a two-way exchange. So I'll give you a sense of what the field work is like that we do, because we've been focused on studying populations that are in more rural areas. They're practicing more traditional lifestyles. And I'm going to explain to you in a moment why we're interested in that. And so we have to bring all the supplies with us to do the lab work. We were really careful, really from the start, to do this research in an ethical manner. And I think that's why we continue to do it today. So back when I started this, not everybody even went through IRB review or went through ethics review. That was often very rare, but we did that, and it would take a long time to get our permits typically. And we also spent a lot of time talking to the communities, as shown here in Cameroon. This is one of my collaborators from the University of Yaoundé, Alfred I'm going to say his name wrong, Alfred Njimshe. And we explain the projects and what we're doing and why we're doing it and what are the risks and what are the benefits of anything, and addressing concerns. And it's only after we get community consent and then ultimately individual consent that we proceed with the study. The other thing that we think is really important is to return results to participants. So we try to do that whenever possible. And I could tell you that based on my experience, people really appreciate that, because otherwise, when I actually, not this group, it was the Sandawe that I went, one of the first groups that we studied in like 2002. And when we got there, I remember them asking, they literally thought that we were coming back to tell them the results of a study that had been done in the 1950s. I mean, they were literally waiting to hear the results of this, and were very disappointed when I said, no, I'm sorry, we're not, I don't know anything about that. So I think it's a type of benefit sharing, and it's really important to do. I think training and capacity building is obviously also very important. And I've had the honor of being able to train a number of grad students and postdocs from Africa in my lab, and continue to do so. So in these regions, we collect blood, and from blood, you can get DNA and you can get RNA and plasma. We collect fecal samples to look at the gut microbiome. We collect information, detailed information about ethnography and diet and so on. And then we have to process these in places typically without electricity. What we started to do is just bring the generator, and then we can just set up the lab in the bush. And when we look at phenotypic variation, this is whatever you can do in a really remote regions without being overly invasive. So very detailed anthropometric phenotypes, height and waist circumference and limb length, cardiovascular, lung and blood phenotypes, like blood pressure and oxygen, hemoglobin, metabolic function, lactose tolerance, glucose tolerance, for example. and infectious disease status when we can. And then we're using this integrative omics approach to analyze the data. So integrating genomics, which at this point we're moving into looking at whole genome sequence variation, proteomics from blood, epigenetic variation, looking at methylation patterns, for example. We look at gene expression from whole blood. We also are doing single cell studies from white blood cells to look at immune response in different populations. We study the gut microbiome, the blood metabolome, and how these are influencing each other and also are influenced by environmental factors like pathogen exposures or diet. How together those are influencing normal variable traits and disease risk. So one of the reasons we have been studying the rural populations is because we want to contrast what we're observing with populations living in more urban regions. If we compare people, let's say, of similar genetic ancestry living in a rural versus an urban environment, we may be able to help disentangle some of the genetic from environmental effects influencing traits of interest. Or you could do the opposite. You could look at groups that have different ancestries, as shown by this, the different colored lines here. This is the inferred genetic ancestry, but living in a similar environment and have different disease risk. Let's see, as some of you may know, are traditionally nomadic pastoralists, sort of going up from Sudan across the Sahel to Western Africa. And for reasons that are not clear, they have relative resistance to malaria. So they don't have like the sickle mutation and other types of known mutations that play a role in malaria resistance. And so people have been really interested in comparing them to neighboring populations that have different genetic ancestries, but they're more susceptible to malaria than the Fulani. The San, as many of you may know, have a higher incidence of TB relative to some of the neighboring Bantu-speaking groups. Again, this could be due to environmental factors, social cultural factors, or could have something to do with genetic risk factors or host pathogen interactions. So this is from a paper we published a number of years ago, but still is does a pretty good job characterizing genomic variation in ethnically diverse populations. So we looked at studied over 3,000 Africans from 121 ethnic groups and used structure. I think some of you might be familiar with that, but a probabilistic approach to infer genetic ancestries, which are shown by the different colors. And we just pooled them by geographic region or by language spoken. And the take-home point is just the large amount of genetic diversity that's seen. So populations in Eastern and Southern and Central and Northern Africa have very diverse genetic ancestries. And if we look, let's say if we look even here in Tanzania, this is over about I'd say about 200-kilometer region, you see so much variation. And this is because of the demographic history that I told you about in Africa. They've maintained bigger population sizes, been very structured, have adapted to very diverse environments. And so for that reason, it's also important to include populations from different regions of Africa. And by no means have we captured all the diversity. So I want to tell you about a study that was published a couple of years ago by Shawa Fan, took a lead in the bioinformatics analysis. And that was together with David Reich and the Simons Diversity, they called it the Simons Genomic Diversity Panel. And we looked at whole genome sequence variation from 94 individuals from 44 African populations. Mainly, this David Reich's approach was to look at just 2 individuals per population. And from that, you can construct a phylogenetic tree. And so many people have shown that the groups that have the most basal lineages in that tree are the San populations from Southern Africa, followed by the rainforest hunter-gatherers from Central Africa, like the Biaka, the Baka, the Betsen. And then we have populations tend to cluster by geographic region and by language. So this is Central and Western Africa. These are groups from some from East Africa, but many from Central that speak languages that originated in Eastern and Northern Africa. And then we have the Hudson and the Sandawe. You can see they're actually closer to some of these other East Africans than they are to the other Click-speaking San. And if we were to put non-Africans here on this tree, they'd be here, basically a subset of the diversity that we see in Africa. So we use this MSMC, which is a coalescent-based approach, to make inferences about changes in the effective population size as you go backwards in time. And you just ignore these more recent dates because this particular method doesn't do very well for recent dates. But if we look at, let's say, greater than 50,000 years ago, what's interesting is that you start seeing divergence around 250,000 years ago. So that's pretty close to the time of origin of modern humans based on the fossil record. And we see that the San and the rainforest hunter-gatherers have maintained the largest effective population size in the past, even though today the census size is pretty small. We can also use this approach to infer times of population divergence. In this particular analysis, the San diverged earliest from other groups, could have been somewhere on the order of 160,000 years, but certainly greater than 100,000 years ago. We see divergence from some of the other hunter-gatherer groups somewhere between roughly 70 to 85,000 years ago. And then we see divergence between populations speaking other language families that again is pretty old, ranging from around 16,000 years ago to around 34,000 years ago. But even between some of these groups that speak somewhat similar languages, the Hadza and the Sandawe, for example, diverged around 23,000 years ago. Two different San groups that speak slightly different Khoisan languages diverged around 30,000 years ago. So you're seeing this history of larger populations and substructured for very long periods of time, but with very complex migration and admixture events. It's a really complex demographic history in Africa that will take, I think, some time to figure out. So I'll tell you a bit about some of the phenotypic variation that we see for some of the traits that we've looked at. And this is work done by Matt Hansen, looking at samples from mainly 3 regions, mainly Central, Western Africa, Eastern, and Southern Africa, genotype for around 2,000 people using Illumina arrays. And these populations practice very different subsistence lifestyles. So people who are hunters and gatherers, and this somewhat depends on where they live, of course, but tend to have a diet that's heavier in things like tubers and nuts and fruits, and then to a lesser extent meat. Pastoralists, again, depending on where they are, have a diet that's heavier in milk, dairy, blood, and meat. And agriculturalist groups will have a diet that's very heavy in grains and maize, vegetables, fruits, and tubers. And we're interested in how people have adapted to these different diets and how does that diet influence normal variable traits. So if we start by looking at things like looking at height, for example, and this is from women, and the different populations are color-coded based on their subsistence pattern. And we could see that at the very low end of the height spectrum are the Baka, Central African rainforest hunter-gatherers, known to be very short-statured, and then one of the San populations. And you tend to see the hunter-gatherers on the lower end. And on the higher end of height, we see actually the Herero, and then also some East African populations. For BMI at the very low end, we see one of the East African pastoralist groups. They tend to have a tall, thin body type. And then we see the San and some of the other hunter-gatherer groups at the low end. And at the high end, mainly food-producing agriculturalist groups, which isn't a huge surprise, I would say. Waist circumference, percent body fat shows a similar trend, tends to be lower in hunter-gatherers, higher in the food-producing populations. Percent body fat is highest in the Herero, which is a population I'm sure many of you are familiar with. And as you may be aware, for cultural reasons, it's considered desirable for women to be very heavy. It's considered a sign that they're healthy and able to have children and so on. And so there could be cultural reasons why we see these different trends. And then if we look at metabolic trait variation like C-peptide or glucose levels, again, we're seeing a similar trend, tends to be lower in the hunter-gatherers and higher in the food-producing groups. So then what Matt did is he inferred genetic ancestry, which is indicated by these different colors. And he wanted to know how, he then wanted to look at these traits and look at what proportion of the phenotypic variation is explained by genetic variation and how much is explained by environment. But he broke the genetic variation down into two parts. He was interested in the contribution of genetic ancestry versus within population variation. So when you look at this, let's look at skin color, for example. The total height of this bar would be the proportion of the variance in the phenotype due to genetic variation. Okay, so think about that as you're familiar with the heritability. And if you don't see a bar, that shows that there's a strong genetic component, this open space. And then this is subdivided into the orange is reflecting the variation that's correlated with ancestry, and the green is variation that is variable within populations. So first, as not surprising, we see that skin color is highly heritable. It has a strong genetic component, as we know, and it's highly correlated with ancestry. If we look at some of the other anthropometric traits, they are also correlated strongly with genetic variation is playing a major role in those traits, not quite as strongly correlated with ancestry. A lot of within population variation contributing, particularly for some of these traits like cholesterol, LDL, HDL, triglycerides, but for some of the other traits, we're not seeing as strong of a genetic contribution. So this is from a study that we did with Chuck Barant at the University of Michigan, looking at lipidomics, so doing mass spec to identify the different lipids in populations and using principal components analysis to see how individuals cluster based on lipid profiles. And here we can kind of distinguish people who are practicing a pastoralist lifestyle and maybe some of the agriculturalists, but there's a lot of overlap there. And if we look at Cameron, we can distinguish the Fulani pastoralists, but again, there's a lot of overlap here. And we wanted to try to distinguish what's the role of genetics and diet on lipid profiles. So if we look at, for example, these populations in Ethiopia, and we're showing the inferred genetic ancestry. So the Weto are foragers, the Kimont are agropastoralists. If we look over at the right, we can distinguish certain lipids that are significantly different between the groups. So this is showing the relative levels of the different lipids. Now the idea is, okay, these guys have pretty similar genetic ancestry, but they have a really different diet. So when we use principal components analysis based on the lipid profiles, we can really easily distinguish them. And that would suggest that the diet is playing a really important role. And likewise, if we look at the Shabu and the Mursi from, again, from Ethiopia, the Shabu are foragers, the Mursi are pastoralists. We can easily distinguish them. They have similar genetic ancestry. So that would suggest that diet's playing a really important role. But then there are some other groups like the Hamer and the Mursi who have somewhat different genetic ancestries, but are pastoralists. They're both living they have similar diets, but again, we can distinguish them. So does that suggest that genetics is playing also an important role? So to try to further distinguish that, together with Rangman Hangzi Lee, we integrated the data with genotype data, and we look for genetic associations. And we identified hundreds of lipid-associated genetic variants. Most of them are actually in non-coding regions of the genome, so they're likely to be regulatory. Many of them have not been previously reported, though they may be near previously reported associations. And that's because given the lower levels of linkage disequilibrium in Africans, you can find map better in those populations. And the strongest associations were replicated in the Cameroonian data set. Now, what was kind of cool is when you dig in a little bit, So let's say, let's look at this one lipid right here. And we do a GWAS and we find that there's a locus of major effect. There's some other loci that seem to be impacting as well, but this is one of major effect. And let's look at this variant right here. And we could see that if we compare the overall levels of this particular lipid between the Shabu foragers and the Mersey pastoralists, that it's significantly different. But if you look within the populations, genotype is having exactly the same effect. People who are TT homozygotes have a higher prevalence of the lipid within populations. Those who are CC homozygotes have lower levels. So I guess the answer is that both are really important. So diet may be contributing perhaps more to between population variation, but genetics is still playing a really important role in within population variation. So the last thing I'm going to tell you about today is our study of the genetics of skin color in Africa. So we were interested in skin color. I told you that I have a background also in human evolution, and we're interested in anthropology, and we're interested in how people have adapted to different environments during human evolution. So in this upper panel, these blue dots represent the melanin levels. Melanin is the dark pigment in skin in different populations, and you could see that it correlates very well with UV levels. So the idea was that when modern humans left Africa and went to higher latitudes, that there would have been selection for lighter skin color, possibly to promote synthesis of vitamin D, which is synthesized in the skin in response to UV light. But in regions of high UV, there'd be selection for darker skin for protection against skin cancer, for example, and perhaps to prevent degradation of folic acid. So the way that we infer the pigmentation levels is you use a spectrophotometer and you shine a light under the arm, which is a region that isn't getting as much exposure to sunlight. And then you can look at the wavelength of the reflection, and from that you can infer the melanin levels. We see a big range, with the most lightly pigmented people in Africa being the San populations from Southern Africa. And then the most darkly pigmented being the Nilo-Saharan speaking populations that originated in South Sudan, who are amongst the darkest people across the globe. Now, this variation that we're seeing is not due to recent admixture with Europeans for non-Africans, for example. This is naturally, most of this is naturally occurring variation. I would say, considering, however, that in Ethiopia, some of the Semitic populations historically have had some non-African admixture. So I guess that's an exception. So then we did a genome-wide association study with a relatively small number of people, only 1,570. And as you know, nowadays people are looking at tens of thousands of people for GWAS. But when you have a trait that's not quite as complex as, say, blood pressure or diabetes, and you have so much variation, phenotypic and genetic variation, there's a lot of power. And so we found 8 independent associations in 4 regions of the genome, and I'm just going to step through those. So the strongest association was at a gene called SLC24A5. There's a non-synonymous variant, an amino acid substitution, that's associated with skin pigmentation. And in fact, it happens to be exactly, and well, actually, before I get there, let me say that in these plots, the variants associated with light skin are shown in blue, and the variants associated with dark skin are shown in this sort of fuchsia color. So we could see that the variants associated with dark skin here are common in Africa and populations of recent African ancestry, like Afro-Caribbeans and African Americans. And then also in South Asia and Australo-Melanesia, and in this case, actually in Asia as well. And then the variant associated with light skin color is at almost 100% frequency in Europe. It's common in Pakistan, some populations in India, and it's common in East Africa. So it turns out this is exactly the same variant has been already identified in European populations. In fact, it was the very first, I think, that was identified to play a role in skin color in Europeans, light skin color. So we wanted to know, is the variant that we're finding in Africa exactly the same? Was it introduced by migration from outside of Africa, or did it arise independently in Africa? So to do that, we look at haplotype networks. So haplotypes are simply how the different variants are arranged in some short region of the chromosome. In this case, we're looking over about 70,000 nucleotides. Each circle is a haplotype. The size of the circle indicates the number of people who have it, and the colors represent the ancestry, or the self-identified ancestry of these individuals. So we can see, for example, that this non-synonymous variant is on a long haplotype that's very common in non-African, mainly in non-African populations. And others have shown that this is probably due to very strong natural selection, that this has risen to high frequency. But if we look at the East Africans, they had the variant on the same exact haplotype background. So what that suggests to us is that it was introduced by migration back into Africa. We think that occurred at least 5,000 years ago. The second association was at a gene called MFSD12. Now, at the time, it didn't have a name. It actually was just open reading frame, whatever. It got a name as we were in the middle of our study based on homology to this group of transmembrane transporter proteins that facilitate transport of ions and amino acids and peptides and so on. Now, the only clue that we had that maybe it has to do anything with skin pigmentation was studies of patients with vitiligo who have, and they compared the RNA that was expressed in the lightly pigmented skin and the darkly pigmented skin. And they found that this RNA levels for this particular gene were significantly different between the two, even though at the time it didn't have a name. And then our collaborator, Elena Owancia from Brown University, confirmed these results and showed that this gene is highly expressed in melanocytes, which is the cell type which has contains melanosomes, which is where the pigments are produced. So if we look at the at this region, we actually found two independent associations. So one of them are markers that are in almost complete linkage to disequilibrium. There is one non-synonymous variant, but they're also near CTCF binding sites, so we really don't know what they're doing. We don't know what the variants are that are impacting skin color. It could be influencing splicing, it could be regulatory in nature. But we could see that the dark associated allele is really only present in Africa in populations with recent African ancestry. Now, by contrast, if we look at this upstream regulatory region, we could see that the light allele is common in Europe, in Asia, parts of India, East Africa, and very common in the sun. And the dark associated variant is common in Africa, South Asia, Australia, Melanesia. So together with our collaborator, Kevin Brown at NIH, he had generated a transcriptomic data set from 106 melanocytes. Remember, these are the cell types in skin that are going to be where the melanin is produced. And 15 individuals in that data set had high African ancestry. So Derek Kelly, a grad student in my lab, looked for correlations between the level of African ancestry and the levels of gene expression. And in this case, high African ancestry is correlated with low levels of expression. And that fits, it's very consistent with what we observed from the GWAS study, in that the C variant is that is associated with darker pigmentation is also associated with lower gene expression levels. Marcia Beltram, a former postdoc in the lab, used luciferase expression assays to show that the upstream region was acting as a very strong enhancer. So it's influencing gene expression. And then our collaborators, Mickey Marks and Shanna Bauman, knocked out this RNA and the protein in mouse melanocytes. And you could see that when they knock it out, you see a decrease. Let me see. No, I'm sorry. You see an increase. So this is the knockout here. You see an increase in the melanin levels, which is similar to what we were seeing in the human data. But what was a little strange to them is that the protein did not co-localize with other proteins in the melanosome, it co-localized with proteins in the lysosome. So still at that time, at least, was a bit of a mystery of how this is impacting development of pigments in the melanosome. And then our collaborators at NIH, including Bill Pavin, used CRISPR-Cas9 gene editing to knock out this gene in a mouse model. This is in an agouti mouse. And we could see that it has a pretty dramatic impact on the pigmentation. So this mouse, which is normally sort of a red, yellowish color, you see that we now see this gray color. So that red yellowish color is actually caused by pheomelanin. So there are two different types of melanin or pigmentation. There's melanin, pheomelanin. And it turns out that this gene has opposite effects. So when melanin's high, pheomelanin is low, and vice versa. Now, what was also kind of interesting is that our collaborators at NIH, when they knocked the gene out and they looked at the phenotype in the mouse and they said, you know, that looks kind of familiar to us. We've seen this before. Well, I heard that one person here is working at the Jackson Labs, and the Jackson Labs has been studying mouse models for many, many, many years. So back in 1948, Mrs. Susan Sobey identified this mouse, and then they started breeding it. And it's called the grizzle, I believe. And so then in the 1990s, there was an attempt to map the mutation that was causing the phenotype. And it should be noted, there are other phenotypes in addition to pigmentation that are being impacted. So they mapped this to about a 2 centimorgan region, syntenic to MFSD12, to the region that we had found. So Stacy Loftus at NIH obtained these mice, and she sequenced the genomes of the mice. And the only region in which she found a difference was a 9 base pair in-frame deletion in exon 2 of the MFSD12 gene. So I was really excited by this because it was one of the rare examples where we used a human GWAS to actually map a mouse mutation and not the other way around, which is how it's typically done. Then together with our collaborators, Yoon Song and Ethan Jewett, we constructed gene genealogy using coalescent approaches. So think of this, if you unwrap this, you could kind of think of it like the phylogeny, but at the ends of these branches these are all individuals, and they're color coded by their population of origin. These sort of blues and purples, as you could see, are non-Africans, and then the Africans are shown in like reds and oranges and so on. And if they have an open circle, it means that this lineage has the light associated allele. And if they don't have that open circle, then they have the dark associated allele. And they looked at the age of the different mutations. So one thing that was interesting is that the ancestral allele is the allele associated with light skin color, not dark skin color. And if we look at the age of the derived allele, that's the dark associated allele, it's almost 1 million years old. So the light and the dark alleles have been segregating in Africa for a very long time. The third association was near a gene. It was about 40,000 base pairs from a gene called DDB1. DDB1 plays an important role in DNA repair after exposure to UV damage. Children who have mutations in this gene have a disease called xeroderma pigmentosum. They can't easily generate pigment they can't easily repair DNA after exposure to UV, but it's not clear what that has to do with variation in skin color. But it turns out that this gene is actually the gene that plays a role in the color of tomatoes. I thought that was kind of interesting. So it is playing a role in pigmentation, but we didn't know what it was in humans. Some of our recent work actually shows that there are other genes in this region that might be playing an important role. But basically we found, again, these 2 independent associations, but all of these are in predicted regulatory regions that are upstream of DDB1. And as I said, we're now doing some other work to look at other genes in the region that might be important. Now, in this case, high African ancestry is correlated with higher levels of DDB1 gene expression, and the dark associated allele is correlated with higher levels of gene expression. And again, we show that these are very strong, acting as very strong enhancers. So then we use some tests of neutrality this particular test of neutrality called Tajima's D statistic to see if we want to know if we see a signature of natural selection in this region. So this is based on the allele frequency distribution. So think of it this way. These are the number of individuals in a population that have singletons, the number that have doubleton variants, tripleton, and so on. And this blue line shows what you would expect under a neutral model. You expect actually to see an excess of rare variants, probably because of our demographic history. But if you see a really strong excess of rare variants, that could be consistent with positive directional selection. And if you see an excess of common variants, that could be consistent with balancing selection. Now you have to compare it to the rest of the genome because demographic history could also result in some similar patterns. So we have to compare it, and we show that it's at the extreme of the distribution that we would see across the genome. So we looked at some of the 1,000 genomes populations and some of the African populations that we studied. And what we saw was this really strong signature of selection over hundreds of thousands of base pairs. We see this negative Tajima's D value very strongly in Asians, East Asian populations, but also in Europeans, so suggesting positive selection in this region. And it's even more striking if you just look at the heterozygosity, the diversity level. It's like wiped out in Asia. There's no diversity. That's what you expect if a variant really rapidly rises to high frequency and it drags with it the neighboring variation. You see a reduction in variation. And also in Europe, we see some, it's not quite as striking, but when you compare to Africans, you could see it's very different. And when we look at the haplotype network, this is over 200,000 base pairs. We see this common long haplotype in non-African populations. And when you look at the gene genealogy, you see this rapid coalescence of lineages from mainly outside of Africa that coalesce around 60,000 years ago, corresponding with the time of migration of modern humans out of Africa. So this might be an example, a rare example of a nearly complete selective sweep, that when this variant was introduced out of Africa, something was very increase the fitness of the people who had it, and it swept to high frequency. It may have had nothing to do with skin color because these genes have pleiotropic effects and there are many genes in the region. So we don't know what the selective force was. And then the last region was OCA2, HER2, which is well known to play a role in pigmentation. So for example, mutations in OCA2 cause many of the common types of albinism. And there had been other variants identified in HER2 that were associated with light skin color and blue eye color, for example, in Europeans. But we found independent associations. And these two we showed were in enhancers that influence the expression of OCA2. And then there was this mutation in OCA2, which is a synonymous mutation in exon 10, and it was not correlated with gene expression, but it was correlated with alternative splicing. So individuals who have the light associated allele have a shorter transcript, and that transcript is exactly missing exon 10, but it's still in frame, still makes a protein, but that protein is missing the transmembrane region. And so it's going to be really altered in its function. And here we see some signature of selection in Europeans, positive selection. But in OCA2, in some of the African populations, we actually see evidence of balancing selection. And if you look at the gene genealogy, you look at the time to most recent common ancestry, it's really, really old. This is about double what we would typically see. And that's kind of a classic signature of balancing selection, that the different variants are being maintained for a very long period of time, but we don't know why. So in summary, in half of the cases, the ancestral allele is associated with light skin color and not dark skin color. And the age of the derived mutation predates the origin of modern humans in most cases. So these have been segregating in Africa, both light and dark alleles, for a long time. What is the implication for the evolution of human skin color? Well, this poor chimp lost all its body here. You can see it's relatively lightly pigmented. It is actually thought that, from anthropologists have speculated that this would have been the ancestral state. and that when our ancestors left the forest and went into the savanna, there'd be selection for more darkly pigmented skin. But I would argue that there could have been variation in Africa for millions of years for skin color, depending on where people were living. There could have been quite a bit of variation. And then what's also really cool is that the only other region that we saw the darkly pigmented allele is in South Asia and Australo-Melanesia, two regions where we see darkly pigmented people. So anthropologists had speculated that maybe the mutations that are associated with dark skin color in those populations arose independently from in Africa due to what's called convergent evolution. But we showed that they're on the same haplotype background as the dark associated variant in Africa. So they were introduced by migration out of Africa along the sort of southern route into South Asia, Australo-Melanesia, and then they've been maintained there because they're protective against UV and they're adaptive. So to conclude, there's an urgent need to include ethnically diverse people in human genomics research. We need to better understand how and why the genetic architecture of complex traits differs among ethnic groups, and to better be able to transfer polygenic risk scores. That's going to require integration of diverse data sets, and evolutionary genetic analyses are informative for identifying functionally important variation. So I just want to thank the funders of this research and the many collaborators in the Tishkoff lab and at many different universities in Africa. And with that, I will stop and address any questions that you may have.